In recent years, a revolutionary vaccine technology has transformed how we respond to infectious diseases. Today, we'll explore mRNA vaccines delivered by lipid nanoparticles, a platform that's changing the landscape of modern medicine. The COVID-19 pandemic presented an unprecedented global health challenge in late 2019, and by early 2020, the world was facing a crisis that would claim millions of lives and disrupt societies worldwide. Traditional vaccine development typically takes 10 to 15 years, but we didn't have that kind of time. What happened next was extraordinary. Within less than 1 year from the time the virus was sequenced, multiple highly effective vaccines were developed, tested, and authorized for emergency use. By March 2026, more than 13 billion doses of COVID-19 vaccines have been administered globally. A significant portion of these were mRNA vaccines, specifically the Pfizer-BioNTech and Moderna vaccines. Even as the virus evolved and new variants emerged, the vaccines continued to provide strong protection against severe disease. This speed was possible thanks to mRNA LNP technology, messenger RNA packaged in lipid nanoparticles. Unlike traditional vaccines that require growing viruses in eggs or cells, mRNA vaccines can be designed on a computer and manufactured using a standardized, scalable process. So to understand mRNA LNP vaccines, it is first necessary to clarify what mRNA is. Messenger RNA, or mRNA, is a single-stranded nucleic acid molecule that serves as an intermediary between genetic information stored in DNA and the synthesis of proteins. At the 5' end, we find the cap structure, a modified guanosine nucleotide that protects the mRNA from enzymatic degradation and facilitates ribosome binding during translation initiation. Following the cap, the 5' untranslated region contains regulatory sequences that enhance ribosome recruitment. The open reading frame encodes the actual antigen sequence, where the genetic instructions for the target protein reside. The 3' untranslated region follows the stop codon and contains sequences that regulate mRNA stability and localization. Finally, at the 3' end, the poly A tail, a long sequence of adenine nucleotides, enhances mRNA stability and translation efficiency. In the Central Dogma, information flows from DNA to RNA to protein. During transcription, genetic information encoded in DNA is copied into mRNA within the cell nucleus. This mRNA molecule then travels to the cytoplasm, where the ribosome reads the mRNA sequence in 3 nucleotide units called codons, each specifying a particular amino acid. Finally, these amino acids are linked together to form the protein. Importantly, mRNA is a temporary molecule. Once it has served its purpose, it is naturally degraded by cellular enzymes within hours to days. Modern vaccine mRNA incorporates several critical modifications to enhance performance. Most notably, modified nucleosides such as pseudoridine or N1-methylpseudoridine replace natural uridine throughout the sequence. These modifications serve 2 functions. They reduce recognition by pattern recognition receptors of the innate immune system, minimizing inflammatory responses, and they significantly enhance mRNA stability and translational output. However, naked mRNA cannot function as a therapeutic agent for several fundamental reasons. First, mRNA is inherently unstable. Extracellular ribonucleases rapidly degrade unprotected mRNA within seconds to minutes. Second, the negatively charged phosphate backbone of mRNA creates strong electrostatic repulsion with the negatively charged cell membrane, preventing cellular uptake. Third, exogenous mRNA can trigger innate immune sensors, pattern recognition receptors such as toll-like receptors, leading to premature inflammatory responses, and mRNA clearance before therapeutic protein expression occurs. These barriers necessitated the development of a delivery vehicle, lipid nanoparticles, or LNPs. LNPs encapsulate mRNA within a protective lipid shell, shielding it from degradation, facilitating cellular entry through membrane fusion, and enabling controlled intracellular release. The mRNA cargo resides within the core of the nanoparticle, complexed with cationic or ionizable lipids that neutralize the negative charge of the phosphate backbone. Ionizable lipids possess pH-dependent charge characteristics for functional delivery. They are positively charged at acidic pH, facilitating mRNA complexation during formulation, but become neutral at physiological pH 7.4, reducing toxicity in circulation. So, when LNPs enter the acidic environment of endosomes after cellular uptake, the ionizable lipids become protonated again, disrupting the endosomal membrane and enabling mRNA escape into the cytoplasm. Helper lipids, or structural lipids, are typically phospholipids that provide structural support to the lipid bilayer and enhance the fusogenic properties necessary for membrane fusion and cellular entry. Cholesterol stabilizes the lipid bilayer, modulates membrane fluidity, and prevents premature leakage of the mRNA cargo during circulation. It also fills gaps between other lipid molecules, creating a more robust and less permeable structure. Finally, PEG lipids, lipids conjugated to polyethylene glycol, extend from the LNP surface, creating a hydrophilic stealth coating that prevents opsonization and reduce clearance by the mononuclear phagocyte system, extending circulation time and allowing LNPs to reach target tissues before being eliminated. They also prevent nanoparticle aggregation during storage and after administration. Together, these 4 lipid components form a delivery system optimized through decades of nanomedicine research. The precise molar ratios and specific lipid selections are critical determinants of transfection efficiency, biodistribution, and safety profiles. Now let's examine the complete mechanism by which mRNA LNP vaccines elicit protective immunity. The vaccine is injected into the deltoid muscle, where LNPs encounter muscle cells, immune cells, and antigen-presenting cells resident in the tissue. LNPs are then internalized by cells through receptor-mediated or adsorptive endocytosis, forming endosomes. As the endosome matures, its internal pH decreases from 7.4 to approximately 5.5 to 6.0. At this acidic pH, ionizable lipids become protonated and positively charged, disrupting the endosomal membrane through electrostatic interaction and membrane fusion, allowing mRNA to escape into the cytoplasm before lysosomal degradation occurs. Free mRNA in the cytoplasm is recognized by ribosomes via the 5' cap structure. Then the ORF is translated into the encoded antigen protein. For COVID-19 vaccines, this is the perfusion-stabilized spike protein. The translated protein is processed through 2 pathways. Some protein is degraded by proteasomes into peptide fragments, which are loaded onto MHC class I molecules and presented on the cell surface, activating CD8 cytotoxic T cells. Other protein is secreted or displayed on the cell surface, where it can be taken up by dendritic cells and presented via MHC class II to CD4 helper T cells. This CD4 T cell activates protein-specific B cells, driving their maturation and antibody production. Memory B cells and memory T cells persist long-term, providing protection upon future pathogen encounter. The entire process from injection to detectable immune response occurs over 7 to 14 days, with peak antibody levels typically reaching 2 to 4 weeks post-vaccination. The mRNA LNP platform has demonstrated broad applicability across multiple disease categories, establishing itself as a versatile therapeutic modality. In less than a year, this technology helped turn the tide against a pandemic that claimed millions of lives. Today, that same platform is being engineered to treat cancer, correct genetic defects, and prevent diseases that have eluded vaccines for generations. This is the power of platform science: one breakthrough, infinite applications, and a future limited only by our imagination.